All right. Welcome back, everybody, to the third session in our academy today. I'm Daniel Inking. I'm the CEO of Everflux Technologies, and also we are collaborating, just for anyone who didn't hear in the beginning, we're collaborating with Leighton Morrison on this initiative we call Everflux University to bring you this education today. So our third session today is with Chip-Osborn with Osborn Organics. We're really happy to have him here talking more about soil chemistry beyond nutrients. And just to let everyone know, just a reminder, our title sponsor for this event is Pitmoss. And our sponsor for this session is Gillespie Brands. Gillespie Brands does branding and marketing. They've actually helped us out quite a bit with ours. So if you're looking for an agency, they're really great. So, without further ado, I think we will jump right into the talk with Chip. And Chip, you can introduce a little more about yourself, and let me pull your slides up here.
Sure.
All right.
All right, so it looks like we're good to go. Well, good afternoon. I'm Chip-Osborn. I'm based on the East Coast. I'm out in Massachusetts. I work nationally in the organic side of land management, land care, both public and private, as well as work with Leighton and others, bringing some of the science of soils to the cannabis industry. And it is science. It really is. That's what it's all about. It's not really just hit or miss, where we know so much more now about how to manage in let's say an organic or a synthetic free system than we ever did before. And it's really because science has made that happen for us, has opened up new avenues that we can look at as far as delivering nutrition to any one of a number of plants. And in our case here, we'll specifically be talking about the cannabis plant. Soil chemistry is at the basic of what's happening for nutrients. And we learn to understand a little bit of soil chemistry, and then we're going to talk a little bit about soil testing and why it's important to understand exactly what's happening there. And in the long run, it's going to save you all money and improve your bottom line, because the days of having to spend big money on inputs should be, moving into the past with what we know and what you've heard from Elaine and Leighton about the biological complex and how that is central and fundamental to nutrition. Here's a slide you probably didn't expect to see. One of my projects, professional baseball stadium, and that has had no synthetics for almost a year and a half. That's growing in 98.6% sand. It's a low nitrogen program. It's liquid biologicals. And the microbes that live in the sand are fed with carbon and vermicompost. So if this can be done in a situation like this, where we can remove water-soluble synthetics. This program this year came in $1,800 under budget, so $1,800 less than they had budgeted for synthetic management, and no synthetics, no pesticides have been used in this system. So if that kind of a thing can be done without synthetics, why not cannabis? What we're really trying to do here is to learn to be a little bit diagnostic, and that's what the chemistry, lecture will be about today, how we understand a little bit about chemistry and how we diagnose. It certainly can be done, but I'm going to walk you through a little bit of a more conventional approach to cannabis growing that that focuses strongly on product input. Leighton and I talk at length about strategies that we can begin to develop that will allow the cannabis industry to produce higher quality bud at a lower overhead. So reducing overhead and focusing more on the natural system. So in a conventional program, this is product input focus. So I want to just show you the intensity, and I'm probably not telling you things that you all don't know, but just as review from the conventional perspective, we look at the early vegetative stage, mid-vegetative stage, late vegetative, and we're constantly juggling fertilizer formulations, starting off at lower nitrogen, moving up to higher nitrogen, backing down on N a little bit and beginning in late vegetative to increase phosphorus a bit. So, we know what the plant needs. We all know that. We all know that it needs nitrogen in the beginning and phosphorus and potassium, a little bit later on. But you can see this is very calculated on exactly what's there. This is called a feeding recommendation. And a lot of you probably have said that it is time to feed the plant. It's time to feed. It's time to feed the plant. And, for this particular one, it is called a feeding recommendation. Now you see we're in the bloom stage, early, mid to late, and late, where we're, again, juggling fertilizer formulations, again, lowering nitrogen up to 10% potassium, mid-bloom, high potassium, high phosphorus, and then late bloom, dialing it back a little bit, keeping potassium up there, and then the pH balance flush at the end. So that's a pretty intense, program that is literally changing very frequently. So in those two slides, why do you think maybe I put the word feeding in quotes? It is in there that way because we have to get beyond thinking about feeding the plant. Because if we are relying on synthetics, we are using water-soluble materials that directly make an impact on the plant. In other words, they can be processed by the plant in very short time because they have been solubilized by moisture. But if we're really trying to grow in a chemical-free, organic, regenerative way, we have to focus on feeding the soil. So we have to put that idea that we're going to feed the plant and then realize that we're really not doing that. We are using organic inputs, or we're using compost, or we're using compost teas that are supporting and enhancing the soil and functions within the soil so that the soil will in turn deliver nutrients to the plant. So that's the concept. The first biggest concept is that feed the soil, not the plant. So, a little bit about me. I'm a professional horticulturist for 47 years, even actually a little bit longer now. I started off in commercial greenhouse production. So I was a commercial greenhouse grower, ornamental horticulture. I was a pesticide applicator. I managed with synthetics for 25 years until I realized, and my focus always was feeding the plant. I have to grab some fertilizer and feed the plant. In the mid-1990s, I realized that really wasn't the right way to be approaching things. And I took my horticultural knowledge and began to teach myself a chemical-free approach, and eventually turned my entire greenhouse range into non-synthetic. So the idea that it can be done. 20 years ago, I moved out of the greenhouse industry into the world of land management and now work in many different areas. One, very closely related to this, and you'll see some soil tests later on, in pasture management. Pasture management, where we're trying to grow pasture grasses of high quality and good density to support grazing animals. So whether I'm doing a pasture, whether I'm doing that baseball field, or whether we're talking about the cannabis plant, we're now always going to think about feeding the soil. So let's look at the growth cycle of cannabis and how conventional overrides that. or how it dovetails with it with nutrition. So we have a seed. So I think we all know that the seed has two things in there. It has a living embryo and it has a package of carbohydrates that acts as energy for that seed. So we plant the seed, we water it in, and the seed germinates. That seedling initially gets its nutrients from that carbohydrate package that's contained within that seed along with the embryo. The seed produces a cotyledon, which is the embryonic leaf, and that leaf has access to those food reserves that are within the seed. There's absolutely no fertilizer at this point, because if you start to stimulate at all with fertility, you will slow down the process of root development. So the plant at this stage absorbs water through the leaves until the secondary root develops. So when that seed germinates, it pushes up a shoot, and it also sends out a primary root system. That primary root system is just that. It's the initial root system, and that is replaced by a secondary root, which becomes the permanent root of the plant. After 3 or 4 weeks, we get 3 or 4 true leaves. We enter the early vegetative stage. Now you can see we're coming in with fertilizers. We're feeding lightly for 1 to 2 weeks with that 2-1-2 or a 4-2-3. 6 weeks after germination, we're aggressively increasing nutrients. Now we're at the 10-5-7s. The higher nitrogen produces our foliage and bud sites. Toward the end of the vegetative stage, we're lowering the nitrogen levels. We're preparing to switch to a bloom booster, so we're now working in the 7-7-7 range. Flowering plants need less nitrogen and more potassium to promote bigger flowers, so hence we're at the 10% potassium formulation. We're dialing up nutrients on all fronts, keeping potassium high. By mid-flower stage, we're at the 6-10-15. Last week of flowering, we're dialing down the nutrients, and then we're beginning to smooth out the transition to that pre-harvest pH balanced flush. So that's pretty intensive from the product input, and there's a pretty significant cost that goes along with that. So if we're going to think about now doing this organically without synthetics and focus on the biomass and balanced soil chemistry, we first need to forget about feeding the plant. We need to remove our focus from product inputs as our driving force. Yes, we're certainly always going to use product input. And I'm going to talk to you about soil amendments and what we might need to do to amend soil based on our soil tests in order to get the optimum nutrients available in the soil for the plant. And our focus is going to be to allow the microbial community to unlock those nutrients for us. We need to think about management of the cannabis crop with a systems-based approach. We can't focus solely on the delivery of nutrients, but we have to look at this plant as part of a horticultural system. Focusing on the creation of a healthy, biologically active soil environment will nourish the plant. If we have, you saw that baseball field that was 98% sand, I have biological life in that 98% sand. It's small. There's not a lot of it there. But with my selection of inputs, I am getting enough going on from the biological complex to carry the nutritional load for that field. So I call it a systems-based approach. And I think that the best way to manage any kind of a crop is to focus on the soil, the environment, and the plant. In other words, that is the system that plant is growing on. The most important thing is we need to focus equally on what is below ground and what we see above ground. Typically, as growers, we are focused on what we see above ground. We make a lot of our applications based on our experience and our visual observation of the plant. And that's, I'm not saying that's inappropriate. That is the mark of a good grower. a good grower that can look at a plant and sort of be one with that plant and understand what that plant needs. And if we approach it like that, I've always said the plant is going to tell me what it needs, as opposed to me following a strict schedule and putting some soluble synthetics in there, literally based on a calendar date. Systems-based management involves three distinct concepts: strong focus on soil biological life, paying attention to proper height of cultural practices. The couple misprints there. The exclusive use of natural organic inputs that help us create the healthy plant and act in a regenerative way in the soil. So we're going to look at and begin to understand soil chemistry. The title of this is Soil Chemistry Beyond Nutrients. That means that if we understand soil chemistry and we learn how it interacts with both soil texture and soil biological life, we're now going to begin to be successful moving beyond nutrients. We're going to learn the importance of soil testing. I'm going to go through some soil tests with you today and show you how they can be used as diagnostic tools. There's no sense putting an input in there if we don't need it. It just costs money. Very often, letting a soil test guide production saves money as opposed to increases cost. We will use inputs, as I mentioned, to adjust soils in relation to soil tests as might be necessary. So the most important thing is that as we learn about soil chemistry, we're going to talk about NPK, and we're going to learn what NPK actually does. We're going to talk about the other aspects of soil chemistry, but then we're going to begin to think beyond NPK. We're going to begin to broaden so that we relate that and integrate that with what is happening below ground. So, conventional view of soil. These next two slides were given to me by Penn State. They are accurate and appropriate for any type of plant we are trying to grow, anytime we're working with soil. So conventionally, we've looked at soils as structural, chemical, or biological. In other words, we have soil structure, which is the relationship of individual percentages of sand, silt, and clay that make up the soil. We have soil chemistry, which will be our strongest focus this afternoon. And that is all of the chemical aspects of the soil, including the ability of soils to hold on to and retain nutrients, as well as the organic matter fraction and how that directly integrates with the biological complex. When we begin to discuss organic matter, we're going to think of that as the home for the microbial community. So the microbes are largely sustained within the organic matter fraction, and humus, that we will discuss a little bit, is one of the driving factors in that relationship. And then we have the biological fraction. These are the microbes. I don't know exactly what Leighton and Elaine, spoke about today, but I will do a little bit of an overview on soil biological life as it directly relates to soil chemistry. But we look at the lower level organisms, the lower trophic levels, the bacteria, the fungi, and then those higher level predators, and how they interact to make nutrient available to our plant. Here's an emerging view of soil health. Modern thinking that, unlike the previous three circles here, that are not related to each other. In other words, we were always taught that structure was one thing, chemistry was another, and biology was a third. And in conventional management, biological and the biology is largely overlooked because synthetic products do not generally require biological intervention to make them work. So we're dealing with sand, silt, and clay, and then water-soluble, chemicals, and that is our conventional view. There's a passing acknowledgement that, yes, there is biological life in the soil, but in conventional management, it is not central to plant management. Where here we have these circles all coming together, meaning, yes, they are very much related. So we learn how they interact. We learn to optimize each area as much as possible. As I mentioned, the organic matter fraction is very important. And we put a very strong focus on humus. Humus as a fraction of organic matter. Humus, by definition, is the end product of decomposition. So when any organic material begins to break down, and if we think of a compost pile, and you put feedstocks in a compost pile, whatever they may be, and they go through a process, and they eventually will turn into compost. A finished compost pile is generally about 60% humus, meaning 60% of that pile has broken down and can no longer break down. It has become stable. The other 40% of that pile is still breaking down. So if that is incorporated in your soils, that continual breakdown is releasing nutrients as the byproduct of decomposition. Compost is not a fertilizer, but make no mistake, compost has significant benefits in the area of fertility. So humus can be less than 1% of the organic matter fraction. So it's a very small fraction of the soil, but responsible for a lot of the activity as organic matter reacts with and supports the microbial biomass. So where these three circles are coming together, right where they all intersect in the middle, think of humus as being the center of that and being the driving factor that begins to pull these all together. So now, our thoughts moving forward is we're going to think about soils as the integration of these three concepts, and then we learn how to manage. Now let's look up here at structural. We're not going to begin to change soil structure. So whatever soil you're working with, whether it is a native field soil or whether it is an engineered in an engineered container soil, we can manipulate structure to some degree. But in a native field soil, we're not looking to try to change that. We're not looking to put a lot of sand in there to break up a heavy clay. We're not looking to put, clay in there to add if we have a very sandy soil. We're going to do a textual analysis soil test that I will walk through you with one, but we will understand what we have there, but we're going to learn to work with what you have as an individual soil, as opposed to coming in and trying to change it. We can work in soil chemistry, and we can use soil amendments to adjust and balance the chemical aspect of the soil to be in the right range for the cannabis plant. And then the biological fraction, as Leighton and Elaine, I'm sure, have told you, we can have very strong influence over that portion of the soil. So soil texture, sand, silt, and clay. So that's a function of the relative amounts of those three minerals. Sand, silt, and clay are all minerals from weathered rock. So different regions of the country, there's different mineral nutrients depending upon the parent rock material. But think of sand, silt, and clay as basically being all the same thing, but they are just defined by particle size. Clay is microscopic and actually is like a flat sheet. Silt is the next largest particle, and sand is a grain visible to the naked eye. Sand can be further subdivided into 5 other textural classifications of very fine, medium, coarse, and very coarse. So this is just what has happened through weathering. But basically, we think of sand, silt, and clay as not three different things, but different particle sizes of basically the same thing. They all contain mineral nutrients, as I said, which varies in different regions of the country. Soil texture affects both nutrient and water retention. So when we're irrigating, texture is going to affect how we irrigate. When we are supplying nutrients, either through fertilizers or amendments, we're going to understand how the texture affects that. Higher clays affect nutrient retention in a positive way. Low clays can affect nutrient retention in a less positive way. These three, how they come together, sand, silt, and clay, will indicate a tendency for the soil to become compacted through heavy irrigation, heavy rain. Just if it's a heavy clay field soil, it is the fine textured particles that have the tendency to compact. So now we look back at that textural analysis, and we know that clay, as I told you, is microscopic. So that's a fine textured particle. Silt also would fall into the category of a fine textured particle. And then, as I mentioned, we have both very fine and fine sands. So when we combine those three numbers together, if we get a total of that soil that is 65 to 75% fine particles, we know that we will have a tendency to compact. Compaction eventually can lead to anaerobic conditions. We'll talk a little bit about that, which in turn will lead to struggling of the root system and a diminishing of the activity in the biomass. We can do soil texture with a laboratory analysis, and I'll show you a couple of lab sheets. We could also do it out in the field with what we call a ribbon test. So you just take a very little bit of soil and moisten it, and you're going to roll it, work it between your thumb and forefinger until you can roll it into a ball. That little ball is, we're not talking a big thing, something the size of a pea or a little bit larger. We're going to roll it into a ball. That ball is called a cast. If we cannot even form it into a cast and it breaks apart, we know that sand makes up 75% or more of that soil. If we can roll that into that cast, then we know we have some percentage of clay there. Then we work it between our thumb and forefingers, and we look to ribbon it out. In other words, to take that little ball and stretch it out to be like a piece of yarn. The further we can stretch that, the higher clay that we have in the soil, because clay imparts stickiness to the soil. So if we can ribbon it out a little bit before it breaks apart, we know that we have clay probably between 3 and 8 or 9%. If we can really get that out to a very thin wire-like ribbon and it holds together, we know we're dealing with clay between probably 12 and 20%. This is the USDA textural triangle. So every soil falls within this triangle. You see right in the middle a loam soil. Loam soil is considered to be the ideal soil for having good organic matter, good nutrient retention, and providing the most benefit to the plant. Go to the lower left and you move into the sandy loams and the loamy sands. To the right, silt and silty loams. You'll see a soil test of a silty loam. And then up in the top of the triangle is clay. And we get into clay loams. Clay loams get to be difficult soils to grow in because bulk density is higher. As you go to the top of that triangle, you get higher bulk density. Higher bulk density means lower air pockets to support microbes. So we take that soil test and then we look at that, and if we have 70% sand and 20% silt, the balance clay, we just simply draw lines across this grid and where those lines intersect determines the textural classification. So, jumping right into soil chemistry now. Soil pH. I'm sure you're all aware of the pH of soils. This is absolutely the first step. Cannabis prefers a pH between 6.0 and 7.0. So we would call that slightly acidic. 7 is neutral. Below 7 is acidic. Above 7 is alkaline. The scale is 1 to 14. And we go below and above 7. But we have to be aware that it is a logarithmic scale. So everything is in multipliers of 10. So that 1 to 14 scale all of a sudden got much bigger. So between 6.2 and 6.3, there's a bigger difference than just 1/10 of a point. So we understand that it is a pretty big scale. And if we're trying to grow cannabis in 6 to 7 pH, and all of a sudden we do a soil test and the pH is in the high 4s or the low 5s, then, we're probably going to be looking at an adjustment. Elaine does talk about, and she's absolutely correct, that if you get the biology right and you get that in the right place, soil chemistry will snap into place. In a lot of my projects, I have to sometimes use a calcium source or magnesium source as a lime to elevate the pH, but I know that if I simultaneously develop the active bacterial fraction, where a bacteria is pH neutral at 7.0, I am going to stabilize the soil and I will keep it there. There will be no more downward fluctuation as long as I have the biomass there managing that for me. So term pH refers to the measure of acidity or alkalinity of a soil, and it is a representation of hydrogen in the soil. So hydrogen is the acidifier, and hydrogen is a cation. Hydrogen can be attracted to exchange sites that we'll talk about in a minute. If there's a lot of hydrogen on those exchange sites, the soil is going to be acidic. We use calcium or magnesium, introduce that to the soil. Either the calcium or magnesium then becomes attracted to the exchange site because the attraction is stronger, and it knocks off the hydrogen. 2 hydrogen combine with 1 oxygen, form water, and work through soil solution, and the pH goes up as the hydrogen goes down. The pH will affect the availability of soil nutrients. At different pHs, nutrients will be made available in different amounts to plants based on species. The pH is affected by the organic matter fraction, by the soil texture that we just talked about, by the presence and numbers of the microbes that are functioning in the soil, the presence and the amount of cations and anions, and the presence of anaerobic conditions, meaning oxygen in the soil. If there is minimal oxygen in the soil, then the pH will change. This is a chart that shows pH and the availability of nutrients. So you can see here when you start to get, the low line for cannabis. So think of cannabis, 6.0, to 7.0. So, medium to slight acidity to neutral. But look what happens if you're in a region of the country where your soils are acidic. And once you start to get down into the 5.5 range, look how nitrogen is restricted. Look at phosphorus, potassium, sulfur, calcium, and magnesium. 6 macronutrients that we will go through one at a time and talk about exactly what they do for the cannabis plant in production. If the pH is not where it's supposed to be, those nutrients will be, they can absolutely be in the soil, and the plant can't process them because they are tied up and not available at the low pH. So look what happens with phosphorus. If you're moving into the, if you're moving from vegetative to reproductive growth, and you are trying to operate in a low pH, and you're counting on phosphorus and potassium to help you, trigger that plant so that the vegetative stops and the reproduction begins. And if phosphorus is all tied up because it can't be used, it's there, you might have applied it. It might be there on a soil test. You might be using a phosphorus fertilizer, but you're just throwing money away because you're putting it in there and the plant can't use it. So monitoring and adjusting the pH through soil testing is absolutely the first step, because if you don't get that correct, all your nutrition after that will just be a coin flip on whether it works. If you are in a region of the country where you have very high pHs, and I have projects where I'm growing at 8.0, but you can see that other than phosphorus, everything else is stable when we get between 7.5 and 8. So nitrogen, potassium, sulfur, cal, and mag can all hold their own and become available at the higher pH. Phosphorus drops off a little bit. Cation exchange capacity. This is a concept that is sometimes difficult, for people to understand the first time through. But I might be oversimplifying a little bit. But it basically, think of this as nutrient retention. Nutrients held in reserve that can be used by the plant as the plant needs it. So cation exchange capacity, generally referred to as CEC, is the ability of a soil to hold basic nutrients, basic meaning a positive charge. So positive charged nutrients can be held on these exchange sites because the exchange sites have a negative electrical charge. So now we're thinking about electrical charge in the soil, and we know what happens with electrical charges. Opposite charges attract, like charges repel. So in this case, we're talking about negative sites in the soil, and the negative sites are in the clay, in some of the clay particles, and in the humus fraction of organic matter. There's actually more electrical charge in humus than there is in clay. I will, show you a soil test that has really high clay, but the nutrient charge isn't that high because that clay doesn't carry a lot of negative charges with it. So CEC is affected by the soil type, the amount of humus, and how they interact together to deliver the framework of electrical charge. Cations have a positive charge. They're attracted. Cations are things like potassium, calcium, magnesium, NH4 plus, ammonium, nitrogen. So when we go through the process of using an organic fertilizer where the nitrogen is in the form of organic protein, or using a compost, when we are having that situation where the active bacteria breaks down that organic fertilizer, it breaks down parts of the compost, it breaks down sloughed off root systems in the soil, and that is converted by the active bacterial fraction. Now that nitrogen from the protein in all of those things is held in the body of the bacteria. Protozoa come along and eat that bacteria and release that nitrogen initially in the ammonium form, NH4. It is in a plant available form because it has now been converted from organic to inorganic. And some of that NH4 is used by the plant, but a good amount of it is held on to in these exchange sites. And think of that as nitrogen in reserve. You are building a reservoir of nitrogen in the soil that the plant can draw on as it needs it, independent of application. Ions have a negative charge, and they're attracted to soil particles, molecules with a positive charge. So think of phosphorus here. Phosphorus is H2PO4 minus. So that is attracted to sands or positive charged particles in the soil. So here is just a little diagram on cation exchange and how plants get their nutrients. So that brown circle there are my nutrients that are on there. We'll focus on one. Potassium's not up on that one, but potassium is K plus. Nitrogen is up there, is not up there, but it's ammonium nitrogen, NH4 plus. So that's a clay particle or a particle of humus. Opposite charges attract. What we have, look at the root of the plant, and we see calcium. Calcium is a second-tier macronutrient that is not thought of initially because we all think NPK, but calcium does a lot of things for us, and we'll talk about what that does. But in this case, calcium is in the root system. There's also calcium in the above ground portion of the plant, but calcium is not mobile. It does not go from root to plant tissue and back and forth, but it's highly mobile in the root system. So if that plant, now you notice calcium has Ca2 plus, meaning it is a strong attraction. Potassium has a single plus, nitrogen, single plus. So what's happening here is this root is saying, I need some potassium from that exchange site. Those calcium molecules become an exudate, and calcium is pushed out of that root hair. It's now in soil solution. It finds an exchange site. It's attracted to the exchange site. It knocks off potassium, and potassium goes back through the root hair into the root to satisfy the need of the plant. So the plant is actually dictating how this back and forth movement goes. So when the plant needs something, it knows it needs to push out calcium and bring back the nutrient it needs. So calcium in soils is always something that should not be overlooked. Soil organic matter. This is probably central to good growing, especially growing in a regenerative and organic manner. It increases the water holding capacity of the soil. You can think of organic matter as a sponge. So you have rainfall or you irrigate and that sponge gets full. And that sponge releases water slowly over a period of time to sustain the plant. When that sponge is dry, we irrigate again. So that's very different than having a heavy clay soil. Any of you that have grown in clayish soils know that water doesn't penetrate very rapidly. That's because it's a fine textured soil and high bulk density. So clay impedes drainage and slows water down. But organic matter is holding it in the root zone and then releasing it to the plant. Air pockets come back into organic matter in the pore space, and then when they fill up with water again, it releases back to the plant. Cation and anion exchange capacity is happening here within the humus fraction. My buzzwords are that soil organic matter is the home for the microbial community. So this feeds the microbes. The carbon, soil organic matter and carbon are the same thing. If we test in soils for organic matter, and if we test for total organic carbon, we're going to get the same number. So soil organic matter is carbon. Carbon is the energy source. Carbon feeds the microbial community. Soil organic matter buffers the pH, can improve soil structure, increases the productivity of soil. As soil organic matter increases, the quality and productivity of the soil increases, therefore the quality of the crop increases. Typically, in agricultural soils, 3% organic matter is considered to be the low line for successful horticultural growth. We typically look to try and establish organic matter somewhere in the 5 to 8% range. If we get, typically not going much over 10, because then we can think of too much of a sponge there and maybe holding water for too long. But increasing organic matter is the number one way to increase the quality of the crop and reduce the dependence upon purchased and supplemental inputs. So soil chemistry, the nutrient pools in chemistry. So all of those things we just talked about are all part of soil chemistry. Now let's look at nutrient and how nutrient for the plant is determined as we look at soil chemistry in general. So over on the left, we have a plant, we have a root, and that root is going to be extracting nutrients from soil solution. So soil solution is organic matter, it's the minerals, it's the air pockets, it's the moisture, it's everything that's in there, and that's referred to as soil solution. So we have inorganic. The inorganic nutrients, that would be something like mineral phosphorus, potassium. Those are water insoluble mineral nutrients that are part of the sand, silt, and clay. So they're water insoluble. They cannot be used by the plant. They have to be chelated or made soluble in order to get into soil solution and be extracted by the root. So once phosphorus is solubilized, potassium is solubilized, it can move into soil solution and the root can extract it. So that is our inorganic nutrient pool. We have the organic nutrient pool. That is with the fraction within organic matter. As protein is being broken down, it is being converted to plant available nutrients, particularly nitrogen. It gets into the soil solution, and then it can be extracted by the root and used by the plant. Then we have the biological fraction, those interactions that happen within the biological community, where one organism is consuming another and making nitrogen directly available to the plant. And that after that biological interaction, that nitrogen and other nutrients move into soil solution and can be extracted by the root. So whenever we're growing a crop, we try to understand what potential in the soil do we have for an inorganic fraction, an organic fraction, and a biological fraction, all to be able to supply nutrient through soil solution to the plant. And how much can we get in aggregate from all of that? So soil testing is going to help us. It's going to help us to understand those three things and what we have for potential there. Once we understand that, then we look at the needs of the crop. So we look at what we decide we want for that cannabis crop and how much nutrient we think we need to have there to take that from beginning to from the beginning to the end to harvest. So we get everything that we can get through the system, inorganic, organic, and biological. Now we have management inputs. So rather than come out initially and say, I'm going to go buy all these management inputs and I'm going to go bang, 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 we really now should think about, boy, I can get this amount from inorganic, this amount for organic, this amount from the biological. I only have to go out and buy what I need to make up the difference from what the soil is currently supplying to me. And that saves money. The inorganic at the top there, which I said was the water insoluble phosphorus or potassium, for example, the biological fraction solubilizes that. The humic acids in the soil solubilize that. Any organic acid acts as a chelator and solubilizes inorganic water-insoluble mineral nutrients so that they can become available. If you buy them as a management input and they're conventional in nature, they have been, and phosphorus and potassium are reacted with acids, hydrochloric sulfuric acid. Acid reacted before going into the bag so that it is water soluble on contact with moisture, where we're using the biological fraction and the natural organic acids in the soil to chelate them. So if I know that I have a lot of phosphorus in the soil, that's a great thing. I just have to know now, how am I going to unlock that phosphorus and make it work for me? Because if it's in the inorganic water insoluble form, it can be there, but it's not going to do me any good. So I have to understand, how do I take that reservoir of mineral phosphorus that's there and work through the biological and the organic fractions to make that plant available, and I don't have to go out and buy it. So that's really how we're looking at this now. We're using soil tests. We're understanding what potential that soil can give me. We're making adjustments if we need to based on the soil test. And we're only going out and spending money on what we have to have. Soil structure is tied in with soil chemistry because it's the way everything functions. But I can't stress enough that this is absolutely one of the most important concepts that I'm speaking about this afternoon. If you have good soil structure, you're 2/3 of the way towards producing a great crop. If soil structure is degraded and you don't have good soil structure, you will struggle because this is central to successful horticultural growth. Soil structure at the most basic level simply describes the arrangement of the solid parts of the soil and the pore space located between them. It's a factor of the size and the amounts of soil aggregates. It's determined by how individual soil granules bind together and the arrangement of pore space in relation to the aggregates. So they can bind together through glues that are produced by the biomass, exudates from the fungal and bacterial biomass that help bind the soil together. Look at the different particle sizes and look on the left. Doesn't look like there's a whole lot of air pockets in there. Look in the middle and you see, good air pockets and then and not so good. And over the right, lots of air, large particles. So, we're typically probably looking at a soil more in the center. But what the most important concept is that pore space contains air. Simply put, all soil particles from the microscopic sheets of clay to the larger grains of sand need to be surrounded on all sides by pockets of air. In other words, particles should not be pressed together, touching particle. That's kind of what happens in a high clay soil because clay is a microscopic sheet and those sheets stack on top of each other and they can reduce oxygen from time to time. Pore space, having those air pockets, oxygen is contained in that air, and oxygen is central to the growth of the root of the cannabis plant and is central to the health, survival, and functionality of the microbial biomass. So the pockets of air, critically important. Structure is developed and maintained primarily by microbes, roots, and arthropods in natural systems. So if we don't have a good microbial population, we're probably not going to have good soil structure. Years of conventional management in any type of agriculture or land management, when it's just been year after year after year or decade after decade of water-soluble synthetic salt-based nutrients, and that's what synthetic fertilizers are. They're just mineral salt. It stands to reason that salt is not conducive to a healthy microbial population. So the repeated use diminishes microbes and degrades soil structure. As soil structure degrades, the plant begins to struggle and becomes totally dependent upon water-soluble nutrient, and it tries to do the best it can with minimal oxygen in the soil. Structure can be influenced by texture, chemistry, and biological life. So again, another interaction in the soil. We have the term bulk density. Bulk density determines how these is sort of a representation of how these particles are arranged. High bulk density, high bulk density meaning the higher weight per ton of when you weigh soil, and you weigh soil when a compost gets up to 2,000 pounds, a cubic yard, bulk density is really high, when a good compost should be about 1,000 to 1,200 pounds to a cubic yard. That reflects a lot of nice air pockets in there. So high bulk density has a negative effect on microbes because the air pockets are reduced. Anaerobic conditions, you absolutely have to be aware of and avoid anaerobic conditions. Anaerobic means the absence of oxygen. Pore space is lost by either compacted soils, overly wet soils, or very high bulk density. Any one of those three things reduces pore space, which in turn reduces available oxygen. What happens when oxygen diminishes and anaerobic conditions take over? Toxic materials are produced in that anaerobic soil. One of those can be alcohols. 1 part per million alcohol can solubilize a plant cell wall. If we get a very anaerobic soil, or if we have an anaerobic compost, either one of those can produce up to 25 parts per million alcohol. So now think of a very compacted soil, or a dense soil, or an overly wet soil. If it goes anaerobic, you build up some alcohol there, you will begin to degrade the root of the plant. So we always want to be aware of anaerobic soil conditions and look for that. A good grower you can smell anaerobic soil. Though as somebody that gets very familiar with soils, you can really, and every time I get on a project and people laugh, first thing I do is pick up a handful of soil, pick up a handful of compost and smell it. You can actually, a good trained nose can smell biological life in soils. So that nice, sweet, rich, earthy smell means lots of oxygen is available there. So now let's jump into the chemistry side of nutrients. Soil is the major source of nutrients needed for the growth of plants. The 3 main nutrients are nitrogen, phosphorus, and potassium. The next 3 in order of importance are calcium, magnesium, and sulfur. Collectively, these 6 are referred to as macronutrients. So NPK are the top tier macronutrients. Cal, Mag, and sulfur, second tier macronutrients. For decades, all growers focused on production agriculture has focused on NPK and largely overlooked the value of the others, because manipulating NPK can produce plant material may not be the healthiest. Soil might not be the healthiest, but you can manipulate through water-soluble methods and do that. So we have our macronutrients here. Let's get little definitions here. Nutrition. Nutrition should be thought of as the supply and absorption of specific chemical compounds needed for normal growth and metabolism of plants. So that doesn't mean always purchasing and introducing, but it's happening naturally or introduced. Nutrients are the chemical compounds that function as raw materials for the synthesis of different structural and functional substances of plants. Very important to think of nutrients as raw materials. Do not think of nutrients as plant foods. They're raw materials that allow physiological processes of the plant to function as they should. Mineral nutrients are the inorganic minerals obtained from soil which are used as raw materials by plant. So that was that inorganic fraction when we looked at the nutrient availability. Mineral nutrition is the absorption, utilization, and assimilation of inorganic compounds or minerals by plants for the synthesis of essential material needed for their growth, development, structure, and physiology. So think of photosynthesis. A lot of times you'll go get it, and there's another slide here, but let me touch on this now. We have materials out there that we can buy, and they call it plant food, tree food, lawn food. Fertilizers are branded as food for a plant. I can assure you that fertilizers in no way are foods for plants. Fertilizers contain raw materials, NPK, and those raw materials are involved in all of these physiological processes. So when we have green in the leaf of the cannabis plant, that's chlorophyll. So if we are using nitrogen as a fertilizer, or we're relying on nitrogen naturally produced by the biomass, that nitrogen, it does many things, but one of the things that it does is intensifies chlorophyll. So nitrogen will make a plant greener because chlorophyll density increases. It is that chlorophyll in the leaf that reacts with energy from the sun in the process of photosynthesis, whereby there's microscopic openings on the underside of the leaf called stomates or stomata. They open and close when the air is the calmest, typically dawn and dusk. And in the case of photosynthesis, carbon dioxide goes right inside that cannabis leaf. Carbon dioxide reacts with chlorophyll, produces carbohydrate, sugars, starches. That is plant food. So the fertilizers have stimulated the process of photosynthesis, which allows the plant to make its own food. An interesting little note on that is that 80% of the carbohydrate that is produced in photosynthesis goes down into the root system. Half of that drives the growth of the root system, and the other half becomes an exudate of carbohydrate into the soil environment. That carbohydrate is now energy for microbes. It is food for microbes. So we are using a fertilizer or natural nutrients in the soil to stimulate the process of photosynthesis to make it as efficient as possible to produce these carbohydrates, which then become an exudate into the soil. So now when we think about regenerative, and now when we think about climate action, and all of those things that are, critical moving forward in agriculture, we are now using the process of photosynthesis to scrub carbon dioxide out of the atmosphere and translocate it to soil-borne carbon, where it becomes sequestered for a long-term energy source in the soil. So I'm actually working, doing a number of tests and on all kinds of landscapes, and I've talked to Leighton about working with cannabis, and we're tracking soluble carbon in soils. And soluble carbon is just that. It used to be carbon dioxide, and it is now carbohydrate carbon in the soil. So we're going to track that over a 3 to 5 year period and try to show that with good, healthy, organic, regenerative growing, we can sequester great amounts of carbon in all kinds of production crops, as well as turf and landscape management, and begin to focus on trying to reduce CO2 in the soils. Essential elements. Mineral nutritional elements are available to the plant as the ion. We talked about that. It's absorbed through the root. Nutrient elements may occur in the soil in the aqueous solution, so in that water portion of the soil. Adsorbed is a word that means attracted to. So it can be attracted to the organic particle, which is humus, or the inorganic particle, which is the clay particle, and that is where CEC comes in. So we can have nutrients that are either freely available in soil solution or adsorbed or held onto these exchange sites. They can be in the form of an insoluble inorganic compound or as a constituent of organic compounds, which can either be plant residues, animals, or living organisms. Macronutrients. This is the top, the six that we discussed, plus that are found in the soil. These are generally present in plant tissue in fairly large concentrations. I have some projects where I do tissue analysis, where I'm not only doing soil analysis, but then I'm regularly doing tissue analysis to determine the effectiveness of nutrients moving from soil through the root into the plant. Total macronutrients, in addition to NPK, Cal, Mag, and sulfur, are carbon, hydrogen, and oxygen. So together that makes 9 macronutrients that are fundamental to plant growth. NPK, primarily obtained from the soil. Cal, Mag, and sulfur, obtained from the soil, needed in a much lower quantity. Then we have micronutrients, also referred to as trace elements. And when we soil test, we are testing for micronutrients. Things like iron, manganese, copper, molybdenum, zinc, chlorine. Manganese is a very small percentage of micronutrients in soil, but manganese is fundamental to uptake of nitrogen by a plant root. So when we look at micronutrients, if we look at manganese and we have low or very low numbers on manganese, we're going to want to consider a minor amendment to get manganese within optimum levels to ensure that nitrogen is functioning as it should and able to easily make its way into the plant. Many of these micronutrients are components or what we call cofactors of enzymes, some essential for electron transfer. Oxygen and hydrogen are obtained from the air, as we know. Carbon can be from the air or from the soil. So in the case of photosynthesis, carbon is coming from the air. In the case of soil, carbon is the organic matter fraction. Nitrogen. It can exist in a soil organic form. It can exist as ammonium, NH4+. It can exist as nitrite or nitrate. We're primarily focused on ammonium and nitrate for inorganic nitrogen that is directly used by the plant. When we have organic nitrogen, which is inorganic matter, which is in an organic fertilizer, the plant cannot directly use that, so it has to be converted to NH4+ or NO3-. What happens when the protozoa consumes the bacteria? We end up with ammonium. We have nitrifying bacteria in the soil that take some of that ammonium and they convert it to nitrate. You'll notice that nitrate is a negative charge. Remember, like charges repel. So this is not attracted to the exchange site. It is water soluble all the time with no ability to be held onto in the soil. Nitrogen is required by all parts of the plant, particularly meristematic tissue, metabolically active cells. Meristematic tissue is referred to as the growth tip, the meristem, where all active growth comes from. It's one of the major constituents of proteins, nucleic acids, vitamins, hormones. Rapid growth increases leaf size, quality, hastens maturity, promotes fruit and seed development. N is the abbreviation for it. Cannabis relies on nitrogen in the vegetative stage for growth and development of healthy green foliage. Seedlings do not require nutrients in the beginning of the process, so we want to avoid nitrogen. Not fertilizing early will help promote the growth of roots, which will benefit the overall plant health in a much bigger way. So when we germinate a seedling, we're not looking to push much nitrogen there at all. Remember that cotyledon. Remember the carbohydrate that was in that seed along with the embryo. That's enough to get it going. So let the plant get going on its own so it can then develop, its root system. Introducing nitrogen at this stage will push the top of the plant and inhibit the underground portion of the plant. Nitrogen can move around the plant while performing other key functions, including photosynthesis, synthesis of amino acids into protein. So nitrogen is mobile, and nitrogen does a lot of things in addition to driving vegetative growth. High or excessive nitrogen applications increase susceptibility of the plant to disease. Root growth is inhibited. It's the first plant to wilt under water stress conditions. Increased potential of insect damage and decreased cool weather tolerance. So as you get, high nitrogen applications can be very, very problematic. So nitrogen is something that, we need to be aware of. We need to understand. And if we are applying it, we are applying it wisely. Phosphorus is absorbed by plants from the soil in the form of phosphate ions, H2PO4 minus, orthophosphate. So phosphorus as P is elemental phosphorus and can't be used by the plant in that form. It can be used when it is combined with hydrogen and oxygen in the form of phosphate. When you look at a fertilizer bag and you see P on the analysis in the middle of that, NPK, that is representing orthophosphate. It's easily redistributed in most plants from one organ to the other, lost from the older leaves. It accumulates in younger leaves, in developing flowers, and seeds. Constituent of cell membranes, proteins, nucleic acid, involved in the energy transfer within plants, activates amino acid production, used in protein synthesis, central to photosynthesis and respiration. Respiration is the opposite of photosynthesis. Photosynthesis makes the energy. Respiration takes that energy and distributes it and turns it into active growth within the plant. Cannabis needs phosphorus throughout the entire growth cycle, but is particularly important for new growth, early stage root development, bud formation, and yield. So it's not strictly flower oriented, because that when that root system moves from primary to secondary, one of the factors that helps that secondary root system get off to a good, quick, and aggressive start is phosphorus. By boosting root growth, phosphorus can help reduce the duration of the vegetative stage. Phosphorus is essential for photosynthesis, metabolism, and nutrient uptake. Phosphorus helps to strengthen and develop strong stems. Phosphorus availability is influenced by pH adjustment. We saw that. At low pHs and high pHs, we can have iron and aluminum at low pH, calcium excessive at high pH. That will lock up phosphorus. If we had a pH at 7.6 and we had high calcium or very high calcium, we could lock up phosphorus with a chemical interaction. Same thing at a very low pH. Iron and aluminum become soluble and interfere with phosphorus uptake. Influenced by the amount and quality of the organic matter, most phosphorus is in the organic matter fraction. So as we improve and develop the organic matter fraction and elevate it from 3 or 4% to maybe 6 or 7 or 8%, we are improving our ability to have a natural reservoir of phosphorus in the soil. The activity of the microbes themselves, phosphorus solubilizing bacteria. When we have that water insoluble mineral phosphorus in soils. There is a specific phosphorus solubilizing bacteria that will interact with phosphorus and make that phosphorus soluble and plant available. Potassium adsorbed as a potassium ion, meaning it's absorbed, it can be used straight as potassium, required in larger quantities in the meristematic tissue, the buds, the leaves, the root tips, and the growth tips. Helps to determine the balance of anions and cations in cell, activates enzymes, critical in the opening and closing of those stomata that I talked about, maintains rigidity, turgidity of cells, thick cell walls, facilitates cell division and growth by moving carbs and sugars between plant parts, and central to disease resistance. So think of potassium in two words: stress resistance. So potassium is something that should be focused on. Is often present in large quantities. Clay particles are the reservoir on cation exchange. It has to be in soil solution to be taken up. And microbes require considerable amounts of K and can compete with, that grass was a misprint, could compete with the plant for available potassium. So we become aware of all of those. So there is K with a strong stem. Potassium helps strengthen the cannabis immune system. So that is the disease resistance. It's strengthening the plant. So it should not be overlooked. Strengthens plant tissue, provides sturdiness, increases the resistance to disease and infections, insect growth, stimulates early growth of the plant, and increases bud size and density. Calcium is absorbed as calcium, Ca++. It's attracted to the exchange sites, adsorbed up here. It works to maintain the stability of soil particles, most notably found in the growth tips, the meristematic tissue. It's mobile, does not translocate between above and below ground, remains in older tissue throughout the growing season, and it can be easily leached. It's involved in all of these plant processes. It is cell elongation, division, germination, pollen growth, activation of enzymes, structural component of cell walls. Most important function is the maintenance of membrane permeability and cell integrity. So even though it's a second-tier macronutrient, you always want to be aware of where calcium is in the plant. Essential to allowing the cannabis to absorb other nutrients. So we saw that, right? Calcium comes out of that root hair and brings nutrients back in there. Absolutely central to that. That's why calcium deficiency leads to other nutrient deficiencies. So we want to have a very keen eye and a soil test in hand to make sure that we are not ever going into a position of being calcium deficient. Because if we become calcium deficient, then we are going to be looking at deficiencies in other nutrients. Gives us better resistance to pests and diseases, protects the plant from heat stress, strengthens cell wall structure, all parts of the plant, stems, leaves, roots, improves the robustness of plant, and benefits overall health. Magnesium, essential for germination. It moves from aging leaves to developing seeds during the life cycle of the plant. It's a critical structural component of chlorophyll, necessary for the functioning of plant enzymes to produce carbs, sugars, fats, an enzyme activator. It's a mobile nutrient, means the plant can relocate it from older tissue to newer tissue. This is why signs of a mag deficiency usually show up on the older, lower leaves first and not on new growth. So if we have that symptom showing up on the older leaves, then this could be an issue. If you don't have the soil test, this is when you should take a soil test. Magnesium plays an important role in converting light into energy. Its presence or absence has a direct impact on the plant's ability to absorb light and subsequently create sugar and carbohydrate. Helps to regulate enzyme production. Sulfur from the soil is the primary source available to the plant. Can also be taken up by the leaves in the gaseous form. It's largely found in stems and root tips of young leaves. It's remobilized during senescence. Senescence is the aging process. Full growth phase from maturity to death of plant tissue. Structural component of amino acid, protein, and vitamin essential to producing chlorophyll. So as you can see here, this second tier macros really can do a lot of stuff. Cannabis plants rely on sulfur to help build the healthy root system, enhance the plant metabolism, support the water and nutrient absorption process, and encourages chlorophyll production and distribution. When growing cannabis, sulfur is available from upwards from a pH value of 5.5. Optimal absorption of sulfur happens at 6.0. Optimal pH, as we talked about for the root zone of cannabis, is between 6.0 and 7.0. So I can't stress enough how important it is to have that soil test that gives you that pH and make that adjustment to get yourself within range. Two major types of soil carbon: biomass, living bacteria and fungi; non-biomass, cellulose, starch, lignin of dead plants. Total organic carbon, referred to as TOC, that is organic matter. It's the carbon that's stored in soil organic matter. Organic carbon enters the soil through the decomposition of plant and animal residues, root exudates, that's from the process of photosynthesis, living and dead microbes, and active and living soil biota. Be careful with carbon additions. Biochar is a great thing, but you have to be really careful because a little bit goes a long way. When we are using humates, which are a form of carbon, and a lot of my projects, when I do a soil biological test and find that I have marginal activity, before I ever reach for a fertilizer, I'm going to reach for a granular humate, which is carbon, because I know that will feed the biomass. But it's a really low rate input. It can be 5 pounds to 1,000 square feet of production area. Maximum rate is 10 pounds. So it's a really low input. And we know that when we are adding carbon or adding humate to soils, there is a maximum amount that we can add in, say, a period of a year when we know it's going to be entirely beneficial. But if we go in excess and put too much in any one shorter period of time, we can have the ability to disrupt the system. Humus, I talked a little bit about. Humus is the end product of decomposition. Chemically stable humus, the importance is focused on fertility in both physical and a chemical sense. Humus has the ability to suppress disease. As the end product of decomposition, the reason it lasts for so long is that it is not decomposed carbon dioxide. So it is protected in the nooks and crannies of the organic matter fraction so that it is not directly exposed to and decomposed. So there becomes that long-term resource. Many, many years, decades, humus can be functional in the soil. Protected from decomposition by microbial or enzyme action because it's hidden inside the small aggregates or it's tightly attached to clays. So that's how we protect it. Humus is one of the most valuable portions of the soil, and specifically soil organic matter, because it is the energy. It is the driving force for the microbial community. So let's take a little bit. Now we understand a little bit about chemistry. We talked about the nutrients. Let's talk about fertilizers and amendments, and then we'll look at some soil tests, and then we'll get into some questions in a little bit. Fertilizer has a guaranteed analysis. So if it is labeled a fertilizer, that means it has a guaranteed analysis. The NPK, 3 numbers on the bag, every bag has that same guaranteed analysis. Soil amendments may have some nutritional value, but they primarily build, change, or adjust soils in relation to soil tests. If you use too much fertilizer material, microbes will not work efficiently for you. So fertilizer is not always the answer. If we are looking at things like mycorrhizal fungi, and I'm sure that you all are aware of Mycorrhizae and what it can do for the cannabis plant by making that root association and bringing nutrients, particularly nitrogen and phosphorus, into the root zone of that plant from well beyond the reach of the root system. Also brings moisture back in there. So mycorrhizae function in a symbiotic relationship with the plant when we have those reproductive structures, associating with the root and bringing in those nutrients. If we go heavy with fertilizer, mycorrhizae aren't going to work. They're going to just sit back and say, I don't need to work. Somebody else is doing all the work. So one of the reasons for beginning to back down on a lot of fertilizer is to let the natural processes take over. If we continually rely heavily on the fertilizer bag, then a lot of natural processes won't begin to kick in. Now, all that being said, there are many, many situations where until we get soil biological life at a healthy point and in the level of activity and relationship to each other that we want, we very well may be dependent upon a fertilizer bag or bottle, whatever it is that you're using, to some degree, to get us going. So we don't always have the time, the opportunity to start planting on day one with a perfect soil. We may have to build soils along the way. So the idea is that you might use fertilizers to begin to feed the soil, which can in turn feed the cannabis plant, knowing that as you are doing this, you are building the soil for the future where these natural resources can take over and begin to do the work for us. Soil testing. So that's the most important first step. So, why do we test? What do we test for? Soil test interpretation and understanding the results. So I'm going to go through some soil tests and then we'll wrap up and talk a little bit about the difference between synthetic and organic fertilizers and what they do or don't do for us. I'm going to talk about 3 different types of soil tests. The first one is nutrient analysis, also referred to as soil chemistry. So this is where all the data points come for all of the soil. Chemistry that we talked about. We get the soil pH, the organic matter percentage, the 6 macronutrients, micronutrients. If we are in a region of the country where sodium is high in soils, we get levels of sodium as well as soil salinity. We get the cation exchange capacity and base saturation. Base saturation is a concept where we have those cation exchange sites and we have those calcium, magnesium, potassium, nitrogen that are held on those exchange sites. So that base saturation will be that measure of what is on there, and we'll show and I'll tell you what will be optimum. The next test is the textural analysis. Sand, silt, and clay, soil's ability to hold nutrients, potential water holding capacity, soil classification, and a tendency of the soil to become compacted. The textual analysis, then when we have those 2 tests in hand, chemistry and texture, now we're beginning to get a picture of exactly what we have there for the physical properties of the soil and what's contained in that soil from the natural chemical perspective. Soil chemistry test, the first one, generally between $20 and $35 or $40, depending upon the laboratory. Make sure when you download a submittal form, you look it over. Some soil laboratories do an upcharge to test for organic matter. In other words, a basic soil test may or may not contain organic matter as a readable result. So you want to make sure that you check the box, because as we've talked about, organic matter is critically important. That's probably the biggest tool that you're going to rely on. And you want to make sure that the soil chemistry test includes the organic matter fraction. So $20 to $40. Textural analysis, somewhere generally between 30 and 75, depending upon the level of screening they do with the sand. So if we want the sand sieved out to all 5 individual textural classifications, then that test is going to be about $75. That's the kind of test I do on that baseball field because it's built as a sand system. So I want to know how much is coarse and how much is fine, and that tells me what is going to happen there. For our purposes here in cannabis production, I think that all we need is a simple sand, silt, and clay. Just 3 raw numbers and not worry about the individual percentages of sand. Soil food web biological analysis. Generally out there in the Pacific Northwest at Earth Fort, I think that the test is about $180, somewhere around there. Back here in the East at Soil Food Web New York, the test runs about $240, $250. This is a biological assay of soil microorganisms. It's going to be our indicator of life in the soil. We will get results on the total numbers of organisms in the soil and the amount of organisms that are actively working for us, along with a lot of other information I will show you. Soil testing is about interpretation. So in a soil chemistry test, all of this stuff here is part of that test. Well, I take that back. Biological life is not part of that sand, silt, and clay. So the top 6 or 7 there. And it really is about interpretation and understanding relationships. There's a relationship between pH and available nitrogens, phosphorus, potassium, calcium, and magnesium. Remember that colored chart that we saw? pH and the availability of nutrients. That's a relationship. Cation exchange capacity, the relationship on how much calcium, magnesium, or potassium, ammonium, nitrogen can be held in the soil. Sand, silt, and clay, how the soil functions. Organic matter, what will it support for biological life? So we learn to understand these relationships and we interpret a soil test in that respect, and then we make our adjustments as might be necessary. I got a couple of tests reversed here, so I'm going to go to this one first. Same laboratory. This is actually, and these are tests that Leighton provided to me on a pre and post amendment of soils, and you can see how things change. And this is his roadmap. I don't think he didn't know I was going to show these tests, and I don't think he's going to mind, but they're really cool because they really show what is going on. So up in the top texture, clay is 9%, 9.13, silt, 21%, sand, 69%. Then this does go through with coarse and fine sand. So this would be a soil, I believe it does say what it is, but it would be textured, it would be classified as a sandy loam. Then we come down to the pH. pH is 7.79, so a little bit high. Electrical conductivity, 131, which means that there is really low nutrient in here. That's the EC is a measure of salt-based nutrients. Organic matter, 4.68%, not too bad. Nitrogen, 818 ppm, a little bit on the low side, almost normal. Phosphorus, in the optimum normal range, which is right about where it should be. Then we come down to, then we do the micro, and, things are all pretty well in balance here. And then we go to the exchange. This is the base saturation, and we're kind of low on what is attracted to the exchange sites. So that was pre-amendment. Now look at an amendment that was made, and, one of Leighton's magic concoctions that he's famous for. Leighton does amazing things. Every word that comes out of his mouth, you should hang on every word because it is a treasure trove of information. He's changed, the clay percentage. So now clay is 3%. Sand has gone up to 84%. He was trying to do a particular thing here. Silt's about the same. pH, same. EC, a little higher. Look at organic matter. He was able to raise organic matter to 7%. That's very good. Nitrogen, he picked it up to 2,500 ppm and brought it up in the high range. Phosphorus also brought it up there. So he's building this reservoir of nutrient in here that he's going to unlock through biological strategies. Micronutrients generally in balance. Good number, like manganese is right up here. So getting all these micros is in a good place. And then he's changed the exchangeable nutrients to some degree so that things are more functioning. Sodium down here, that's exactly where that wants to be. We don't want sodium actively participating in our functions. So that is what happens on that. Oops, sorry about that. Here is a University of Massachusetts back here in the east, and this is a very detailed textural analysis. So you can see we have 63.8% sand, 25% silt, and 11% clay. So this is a soil that is classified as a sandy loam. It's got a 4.6% gravel content, which is particles larger than the larger fractions. And this shows all of the sands being sieved out to all of their individual classification. Here is the soil chemistry test that goes along with that textural test. So going back to the textural test, we've got 11% clay, 63% sand. So that's not too bad as far as what you would like to see. 65% sand is a good place for sand to be, and the clay at 11% gives good nutrient retention. So this all looks pretty good from that aspect there. Now let's look at the soil chemistry side of things. pH, it's in range, a little bit low. But this could be elevated. We could use a calcium or magnesium lime to elevate it. But now look what's happening here. Phosphorus, 1.3 parts per million. It should be between 4 and 14. Potassium, only 81 ppm, and it should be here. Think of this, stress resistance, strong, healthy plant. Calcium, 1,000 to 1,500 parts per million is where it should be. We're at 623. Magnesium is in range. Sulfur is okay. Micronutrients, generally where they want to be. For some reason, manganese is high here. Iron is really high. That can be, this is a New England soil, so that can be a characteristic here. And another characteristic of in New England soils is high aluminum. So aluminum typically wants to be below 75 ppm. This soil has it at 109. Cation exchange capacity, that's not great. A good CEC wants to be between 12 and 15 milliequivalents to 100 grams. So, going back here, look, we had 11% clay. So that means that clay doesn't have much of an electrical charge. Down here, we have 4.4% organic matter down at the bottom. So that's not bad. average nationally is 5%. So it's not too bad. So there's a potential home for microbial populations there. But the message is we're not going to hold on to a lot of nutrients here unless we introduce something with the ability to do that. And humate, a granular humate, will add exchange sites into the soil. So this particular soil needs a lot of work. So soil texture looked really good. When we're looking at soil chemistry, we've got multiple issues here that need to be addressed. If we didn't have this test and we went on and tried to grow something, whether it was a lawn or a cannabis plant, we would struggle here. Let's look at a little soil biologicals. This is one of my pasture projects. So this is growing pasture grasses as a food crop. This is a test that was done at Soil Food Web Laboratories back east here in New York. We see the active fungal fraction, pretty good. It's a little bit above range. So that means there is good fungal activity. And we have good total fungal presence. So total means the sum of the active and the inactive or dormant fractions. So our job then becomes developing a strategy to take as much of that total number as we can and turn it into active organisms that are actually working for us. We look at the hyphal diameter of those fungi. At 3.25 micrograms, we're looking at that as likely being disease suppressive fungi. As the hyphal diameter increases, so does the ability of the organism to suppress disease. The active bacterial fraction is a little bit low. Not alarmingly low, but low thinking. Remember that active bacteria breaks down anything organic, any of the organic forms of nitrogen, whether it is internal in the system or introduced through supplements. Active bacteria does that. So there's probably enough activity here to begin to break down organic matter and convert it to nitrogen, but it needs to be improved. And a humate will do that. Molasses will do that. The total bacterial fraction is twice optimum. So that means the potential here is very, very good. So now our strategy just becomes converting more of that 656 number into that 29 number. The fungal to bacterial ratio is 0.47 to 1. So typically that F to B ratio probably wants to be a little bit higher than that. So we would like to look to increase fungi. The flagellates, going down to the protozoans. Look at the flagellates and amoeba are both in red, 1600 and 3200 respectively. It is the flagellates and the amoeba that are feeding on active bacteria and releasing ammonium nitrogen to the plant. Ciliates are the measure of anaerobic organisms, and they're below range, which means this soil is not anaerobic. But look at the nitrogen cycling potential. So this is a number that over a 90-day period, we could expect this biomass to cycle this amount of nitrogen. And it's less than 25 pounds of nitrogen per acre. So that is a really low number. And the reason that number is where it is because the flagellates and amoeba are so low. And they really need to be changed. That's where compost tea comes in. That's where a compost comes in. That's where carbon in the form of humate comes in. Whatever it takes to... You can't go out and buy flagellates and amoeba as an input like you can bacteria or fungi. So you are relying on that. A high-quality compost tea is the best way to introduce those guys. Here's another food web analysis. Same laboratory, different form. They changed their database. Active bacteria, and it's a different scale. They did the whole different database. But we're looking at a range of 1 to 5 units, and we're at 41.6. total bacteria, we want to be between 175 and 300, and we're at 762. Active fungal between 1 and 5, and we're at 87. Total fungal wants to be between 175 and 300, and we're at 1345, with a hyphal diameter of 3.5. So everything across the board is above optimum, meaning this biomass is firing on all cylinders. Protozoa, look at the numbers of flagellates, 79,000. We want to be 5,000. 192,000 amoeba and we want to be at 5,000. Ciliates are low, meaning there's plenty of oxygen there. So we have a tremendous amount of protozoan activity here that is feeding on that excellent fraction of active bacteria. A little bit more to the right, we have our mycorrhizal colonization. I think I missed it on the other one, but it was a little bit low on the other one, but it was there. But this is mycorrhizal of 24% and growing as long as we continue to provide food sources to feed the soil. Fungal to bacterial ratio, 1.77. It was a little bit high for this crop, but within the range of where it should be for most production. On the bottom right, plant available nitrogen supply. This soil will produce over 250 pounds of nitrogen per acre over a 90-day period, independent of fertilizer input. And this is where we want to be. This on this soil did not happen overnight. This soil was a project of mine that took a couple of years to get to this point. I didn't have the ability to have Layton's compost tea. I didn't have the ability to have that. So I did this the old-fashioned way and did it with soil amendments and the right things and monitoring soil tests and growing microbes as they needed to grow. Had I had a high-quality compost tea, I could have changed this very quickly, possibly within one production cycle. Here's one I want to show you. This is a compost, just to show you that we can do the same thing with compost. I never use a compost without doing a full compost chemistry, meaning telling me exactly what my nutrients are, my carbon to nitrogen ratio, and all of that. And I match that up with this test, which is compost biology. And then I do a third test, which is only done at one location in the country, where I test my soils for carbon, which is at Woods End Research Laboratory in Maine. And they are probably the leading compost consultants in the country, if not in the top 2 or 3 in the world. And they do all my really in-depth compost testing, where I'm spending $650 per sample, and I'm getting every piece of information on that. Or doing a soil test for $95 a sample that's giving me what is called a soil health score and how my soil relates to soils within my same geographic region. Really cool test, and something that's very new, and it has just been developed within the last year or so. So here's this compost sample. You can see active fungi, really good. Total fungi, very good. Hyphal diameter, good. Active bacteria, good. They're all excellent, actually. Total bacteria, huge total bacterial population. So this is a bacterial dominant compost. It's sort of a balanced fungal and bacterial biomass with a strong emphasis on bacterial activity. Flagellates are way above range. Amoeba about half of where they should be. Ciliates are okay, so it's not not overly compacted. Good pore spate exhibit, and this compost will have the ability to produce between 100 and 150 pounds per acre nitrogen over that 90-day period. We don't do mycorrhizal in compost, because in order to get a mycorrhizal organism, we have to have a root association. Let's see. We're going to go through this. We're going to move right through this now. I'm going to give you quick fertilizers. I think we've got maybe about 10, 12 minutes before we go to questions. Organic fertilizers, their minerals, plants, and animal byproducts are the sources of nitrogen. Organic-based fertilizers are some of the above plus biosolids, storage sludge, or urea. In organic production, we are never using biosolids. Biosolids are just too sketchy. They can have heavy metals. Most importantly, residual pharmaceuticals, which can pass right through into a plant. So organic-based materials are those materials when the industry has put fast-release nitrogen in there because people don't have the confidence that organic fertilizers will work. Synthetic fertilizers are the result of the Haber-Bosch process that created ammonia, or of the acid-reacted fertilizers that I mentioned to you. Urea is the poster child for synthetic fertilizers. That is produced in a chemical reaction that involves ammonia, which is 72% nitrogen, carbon dioxide, 3,500 psi at 350 degrees, and urea as a water-soluble inorganic nitrogen is formed. Synthetic fertilizers, the production consumes fossil fuels, takes 5 tons of petrochemical equivalent, natural gas and oil to make 1 ton of 4600 urea. So it's not sustainable from that perspective. Releases greenhouse gases, disturbs the soil ecosystem, high salt, all mineral salt, upsets the balance of the soil ecosystem and the balance of microbes, and it has the ability to leach. Fertilizer nutrients, nitrate, nitrogen, ammonium, nitrogen, potassium, and phosphorus, all found in a fertilizer bag. I mentioned that it's not plant food. Very important concept. Photosynthesis makes the food, and food is always in quotation marks because, it's the carbohydrate is the food. It's not fertilizer. So the nutrients don't provide energy for growth. They are simply catalysts, raw materials. Very, very important concept. Mineral nutrients have to be made soluble. So we've talked about that a couple of times. That when we are dealing with that soil test and looking at what is there for mineral nutrients, we are looking at what we have and how we can function in that system to make that all available. Mineralization is the science behind what makes organic work, what organic nutrition work. So when we have that bag of organic fertilizer, when we have that liquid organic fertilizer, we have to have active bacteria to break it down. The exception in liquid fertilizers are the new nitrogen soy protein hydrolysates, where science has been able to extract amino acids from insoluble soy protein, where soy protein needs to be broken down by a microbe and converted from organic to inorganic. Well, we can now extract that. We can extract that amino acid. It's 100% water soluble, and because it is an amino acid that was produced in photosynthesis, it's already in a plant available form. So when we use that in production, we can spray it on the foliage and it can be foliar absorbed, or we can introduce it to the soil and it can be drawn in as rapidly as a water soluble synthetic, and it is 100% organic. For the organic, other organic nitrogen sources, they're the proteins and grains and animal byproducts that are broken down by mineralization. Sloughed off root system, deceased microbes, returned plant parts to the soil, all are broken down in the process of mineralization. Protozoa eats the bacteria, break it down, Nitrogen's in the body of the bacteria. Protozoa consumes the bacteria, releases the nitrogen because the protozoa does not need it for its own survival, and it's released as ammonium. Then the nitrifiers pick it up and convert it to nitrate. And plants, every individual species of plant has its own preference on the percentages of ammonium and nitrate that it is that is given to it. So conventional fertilizer, natural, organic. Conventional fertilizer, synthetic, inorganic, water soluble, fast release, fast green up, rapid uptake. It can be encapsulated to slow the breakdown down, or secondarily reacted with formaldehyde or other chemicals to actually require microbial breakdown. Feeds the plant. Multiple applications. Leaves the soil quickly. Used to be inexpensive, but now much more expensive because this followed the cost or the price of the petroleum market, of the crude market. So, a number of years ago, petroleum spiked and went from $50 to $100 a barrel, and the cost of fertilizers doubled. Synthetic. They never came back down. So they're still up there. The price of oil's back down. So synthetic fertilizer manufacturers, just because of market fluctuations, have become more profitable. This slide looks like it was a little bit distorted in conversion. But natural organic, as we mentioned, nutrients of plant, animal, and mineral base, is water insoluble nitrogen, WIN. If you buy a bag of organic fertilizer, should be on the bag, meaning microbes need to break that down. Slow release. This is the feed the soil material. Long-term benefit. We're building soils beyond just the crop that we're doing. Yes, we are using these materials to provide catalysts and raw materials to the cannabis plant, but we're also using these microbes to feed, using these fertilizers to feed microbes in the soil to improve the soil for the next crop. And so we're continually looking to improve. So don't think of a soil as you have a soil, you're going to plant a crop, that crop's going to take all the nutrients out of the soil, and then you're going to go buy more nutrients for the next cycle. We're looking at, we're having the crop in there, we're going to supply the nutrients, that plant's going to get all the nutrients that it needs at the right time. And then when that plant and that cycle of crop is done, that soil has improved. We are regenerative. The soil is getting better, it's getting healthier, and it's in a better position to step off with the next crop. Compost and compost tea, compost nutrients, organic matter, microbial life. Compost tea is microbial life, the best way you can get active microbial life. Using compost and compost tea can successfully replace high levels of fertilizer inputs. And that's really, I think, what, this day is really all about, is beginning to learn how we can rely on a functional biomass to get us what we need in the form of plant nutrition and reduce what we have to go out and purchase. So now we're learning to manage through the biomass. So last couple of slides, just to reiterate what I'm sure Leighton and Elaine talked to you about, but with a healthy soil, healthy biological life, with oxygen present in the root zone, all of these statements are true. Nutrients are held in the soil. Water is retained in the soil. Disease does not have a functional place to grow. The eggs of many insect pests become a food source for biological life. Nutrients are cycled at the correct rate that the plant needs, and the plant controls nutrient availability through exudate production. Number 5, nutrients are cycled at the correct rate that the plant needs. So that very first slides I showed you with that schedule of input. Now start to think about, we're going to come up with a system here that, yes, we're going to be doing some inputs, but basically we're going to rely on this biological life to cycle these nutrients for us as the plant needs it. Nutrient retention, water retention, nutrient cycling, building and maintaining soil structure, critically important. breakdown of toxins and pollutants, and disease suppression. Nutrient cycling. The idea that we can cycle 2.5 pounds of nitrogen every 30 days through natural processes to our plants. This is the end goal of a healthy soil. In order to achieve good soil health, good soil quality, we have to get to the point where we can naturally cycle nutrients independent of outside applications. So what I've tried to do today is to give you a basic understanding of soil chemistry. Will help to initially balance the soil through the use of soil testing. So finding out where you are on day one, and then what needs to happen with a basic understanding of chemistry in order to get that soil to a functional point. The strong focus on soil biological life management is the key to successful. It's a key to successful cannabis management by focusing on compost teas, microbes, the inputs that directly stimulate them in a way towards regenerative management to continually improve soil health, which in turn leads to fewer inputs. The end result is reduced production cost. So thanks very much. And Daniel, I'll turn it back to you if there's any questions.
Thanks, Chip. That was amazing. I'm sure you weren't paying attention to the comments here, but we had so many people saying, wow, this is like so much great information like I've never had. I mean, I've personally never had such a thorough explanation of some of these concepts. So that was really great.
Good, thank you.
So let's, so we've got a bunch of questions here. I think we can probably get to a bunch of them. I just wanted to start off real quick. If we can navigate back closer to the beginning here, I just want to ask for a quick clarifier about this chart here and about the different breakdowns. So, I've kept hearing you say, I've heard this many times, soil is composed of sand, silt, and clay. Now, just clarify a little more, where does loam fit into that? Is loam like a particle size that's in between clay and sand? And why is that one not mentioned as one of the soil components?
Loam is considered to be the ideal soil. And loam, it's when you have a very specific relationship between the percentages of sand, silt, and clay. So if we look at that chart and we look over at clay and look over on the clay leg over on the pink there and say, well, just go to the 20. So if we had 20% clay, we're going to draw a line straight across there. Then go down to sand. And if we had 50% sand, you'd draw a line up through there. And then silt, if we were, up at 30% silt, you draw a line. And where those three lines intersect is the textural classification. So if sand is generally between, 45 and 65%, and clay is between maybe 9 or 10 and 18%, and silt is the difference, those ranges are all going to intersect in the center of that triangle, and that soil is now classified as a loam. So loam is a word that is, and I'm glad you brought this up because I probably didn't elaborate a lot, but that a lot of people go out and say, I'm going to go out and buy some loam to work on a project, right? I'm going to buy some loam. I need loam. Well, loam is a textural classification. If truth be known, when you buy in topsoil, you're probably not buying a loam because it's really valuable. Pure loam is so if there is a soil engineer company or a producing company that sells topsoil, they're going to go out and find a good farm loam somewhere, and they're going to stockpile it. And for every 2 scoops of loam they give you, they're going to give you a scoop of sand, and they're going to mix it in there. And they're going to turn that loam to a loamy sand because they've added sand to it. And all the other fractions in here, as you get more clay, then it becomes a clay loam. As you get more silt, it becomes a silt loam. And as you get more sand, it becomes a sandy loam. So out in the Pacific Northwest, you have sandy loams. You have good loam soils. Here in the Northeast, we have sandy loams and loamy sands. So down in the Southwest and Southeast, it's all, sandy loam, loamy sand, and actually sand. So, so loam is a textural classification when the individual percentages align.
Okay, got it. And then just one more question back to the soil tests, so just clarify this a little bit more for me. So, we get the soil test back. Obviously, like organic matter is really important. Knowing the sand, silt, and clay percentage is really important. Then we have the nutrient analysis. And I mean, we know we're not supposed to do that. We know, if you have a calcium deficiency, we're not going to go buy some CalMag and pour that on because that doesn't work. But what do we do with those? If we have a soil test that tells us we have a certain nutrient deficiency, would you say go out and get some sort of organic amendment that will provide more of that, or is there something else we should do with that information?
Yeah, that's the case when you would consider using an organic amendment. We can change things over long periods of time, with just focusing on the biological complex. When biology gets all snapped in, nutrients follow generally. But, in production, we don't often have that luxury. We don't want to go through and waste a couple of crops to get to the third good one. So, yes, so in that case, if you had a calcium deficiency, you would choose a calcium source to amend that, to correct it, right? But you have to look at the pH because calcium can be part of lime, and lime elevates the pH. So if I have low calcium and low pH, I'm going to get a calcium-based lime, and that's going to do the job for me. But if I already have the pH in my target range, and I don't want it to go any higher, but calcium is low, which is a very common scenario, then I'm going to use pure OMRI certified gypsum, because gypsum is a pure calcium source that will positively influence calcium in the soil and not play with the pH. And all of these things, it's little by little. You don't dump a lot, because remember, calcium, magnesium, potassium, phosphorus, it's all still salt. It's all mineral salt. So if I had a magnesium deficiency, I might not touch that. Calcium is important because it's important in soil structure. It's important in microbial health. It's important in that root zone in the cation exchange there, to calcium exit goes out and brings the potassium back in. Magnesium is central to chlorophyll and a bunch of other things, but I would really have to have a severe magnesium deficiency showing before I wanted to put that in, because the only way I can change magnesium is with Epsom salt, magnesium sulfate. So you're trying to build a healthy biomass on one hand, and all of a sudden you want an amendment here that's salt, and you got to put it in there. So you have to use good judgment. And if you have to put salts in, it's always a little bit, and you inch yourself along rather than go, I'm just going to dump the bag in and make the correction.
Great, got it. All right, so let's get to some of these upvoted questions here. So would I be able to soil test at home, or would I send it to a lab? Is there a home soil test, basically?
There are home soil tests, but when we're dealing with production agriculture, they're not something I'd want to rely on, because you can get a very good guesstimate. There are some brand new tests out that are a little more high tech that'll get you. But when these tests, when they go to a laboratory, you take a sample of soil for soil chemistry, and they're going to take that sample and they're going to put an acid in there. And that acid solubilizes the mineral nutrients and allows us to be able to read them and put them in a readable form, parts per million. So and some labs use strong acid, some use weak acid. But the idea is that if you did it at home, you'd get a good guesstimate, where in the laboratory they can drill you right down to very close to parts per million of all of these things. And, when you're doing, cannabis, as we can see, it has the desired pH range. Well, if you're off by a 2 or 3 point on the low side, and the test isn't that accurate, then you might get into a situation through the bloom cycle of having phosphorus locked up. So I'm even though tests are available for home, strong advocate of using laboratory.
Great. All right. So next question, what are your thoughts on putting bands of fertilizer, and I assume this means organic fertilizer, in soil for high feeders? So thinking bone meal or rock phosphate, but in an effort not to harm mycorrhizae because of higher phosphate. I'm not quite sure what that last part means, but I think it's just generally about those, do you recommend things?
No, banding is fine. You would, I would, before I would want to have all my soil tests in hand, including biological. And, for me, I work on all kinds of projects all over the country in different facets of all of this. And I won't work with anybody unless they allow me to do all 3 soil tests, because I can't tell. So if I don't know what the biological complex is doing, and I go in and I band too heavily with a phosphorus, then I might impede the development of mycorrhizal fungi, which I don't want to do. Remember, if you're doing a phosphorus with a black rock or a soft rock phosphate, remember that is not probably going to be immediately available unless you are introducing phosphorus solubilizing bacteria with it. So that is sort of a long-term correction. So I would use black or soft rock phosphate if I had a soil test like that one I showed you where phosphorus is 1.4 parts per million and it wants to be, 6 to 10 or even higher, then I might use black or soft rock phosphate, but I would know that I am building for the future, that it's not necessarily going to be available in large amounts for that initial crop.
Great. All right. And what do you recommend as a good base soil to start off with and then to further amend later on? Any recommendation?
A base soil, are we talking field production or container production?
Good question. The person didn't specify. I would imagine container production or raised beds.
Raised beds or so typically when you engineer a soil, if you have the ability to create your soil, keep the number in mind of 65% sand, right? So 65% sand, and you would like to have between 5 and 10% clay. That's going to give you 2 characteristics: good drainage and nutrient retention. You want organic matter somewhere between 5 and 8 or 9%. So that's going to give you exchange sites for nutrient retention and humus. It's going to give you some clay sites there, and it's going to give you good drainage. So that is when we engineer soils, that's kind of very general, and it can change from crop to crop. But, if we are doing a raised bed or a container, drainage is critically important. And it is the pore space. Again, I can't stress enough, pore space. Having that every particle, no matter how big or small, surrounded on all sides by oxygen pockets. So you have that 65% sand, 10% clay, a little more, and then, 5 to 8% organic matter.
All right. Let's see. What are your thoughts on adding small, frequent amendments compared to one large initial addition?
All depends on what the amendment is. If it's a salt-based amendment, I'm really careful. I'm always going baby steps. Unless I have to try to. See, if I'm going to adjust the pH, that's an amendment. I can do it over time, over 100 days, by using a pelletized calcium source, or I can use a very soluble, finely screened calcium that'll make a push for me in 3 weeks. And then sometimes I might have to do that, but I don't do it in a large amount. I would always go at the lowest rate. You heard me talk about humate as an amendment, 5 pounds, 1,000 square feet. That's a really low rate. So it's a little by little, because what you're doing, unless you're doing a container production where it's a one-off, where you're going to, do the container, and at the end of the cycle, the soil's just gone, you're going to dump it and start over again. That's very different than field production, because when you make amendments in container production, you want to know right off the bat on day one that you've got that where you want it to be. And you should not have to make amendments. I've got processes that I've developed for container production where I can get all my amendments in the pot all at once in the beginning. And certain things happen that they're there. But I'm not going to go halfway. If I have to go halfway through the cycle and say, oh, geez, I got to throw an amendment in there, well, that means I didn't do something right in the beginning. If we're doing field production and we make amendments, we're building that field soil, not just for that initial crop, but for every crop that follows.
All right. So what sample collection techniques would you recommend when testing the soil? And this person's specifically asking about a 10-gallon pot or less, so like a really, really small pot, and wondering if that would disturb the soil too much. But I think generally about soil collection sample technique. Could you just repeat the first part of that again? I got distracted, didn't hear it. Oh, no problem. Mainly the question is about what sample collection technique would you recommend for testing soil? Okay, so in field soil, I'm going to go to 10 or 15 representative areas in my production, and I'm going to go down into the root zone. I'm going to go down probably 4, 5, 6 inches. I'll take some samples, some soil from the 3 to 4 inch range, and then some soil from the 5 to 8 inch range. And I'm going to take all these individual samples and I'm going to compile them into one composite sample. I'm going to mix it all together and form one composite sample. I could choose to test individual production rows. I could do a field. I could do 2. But in some cases, if I'm testing during active growing and I notice I've got a problem, I'm not going to include that soil from those plants into the growing. So I'm going to isolate, right? I'm going to keep things separate and focus. So random, multiple samples combined to a composite, mix it all together. I need about a cup of soil for soil texture, 1 cup for soil chemistry, 2 cups for soil biological life. Soil chemistry, we clean the sample so there's no roots in there, no particles of organic material, because the organic matter test is loss on ignition. Weigh it, burn it, weigh it. Organics burn off, mineral stays. So organic matter or carbon is a representation of the weight of the soil. If I'm testing container soil, I'm going to probe down, and it won't really disturb the root system. If I were testing container soil during production, I would probably be testing because I've noticed a problem and I need to try to correct it fast. Otherwise, container soil should probably be tested before it ever gets into the container. So I know exactly what I have there. And then if I want my amendments, I can make them accordingly pre-plant. So if I'm testing a container in production, that means I've noticed something that's out of whack and I'm looking for a quick down and dirty chemistry so I can come in with something relatively soluble and organic to correct that deficiency and not interfere with production towards the end of cycle. All right. The next question is about the most cost-effective / organic way to add nutrients and amendments. Most cost-effective way would be compost and compost teas. Absolutely. That's where we're going with all of this, because it is the most cost-effective. We may not be able to get there initially. Leighton and I have talked about this at length, that's our goal, is to run the system with basically that. But a lot of times in field production, and if I'm in container production, I can make that happen on day one.
I've worked and developed soils that are all inclusive that will just take that container off and run it right through the cycle with, nothing more than, irrigation with compost tea. Field production is often dependent upon a fertilizer bag initially until we could get the compost and the compost teas ramped up. But the goal is that we don't have to depend upon it all that much. So most cost effective for organic fertilizers, soybean. Soy is very easily processed by the bacterial biomass, and that grain converted into nitrogen very efficiently. Soy fertilizers generally are affordable, not high cost. There's some of the poultry manures. A lot of those, that's probably the least expensive fertilizer. Nothing wrong with it. It grows a good plant. It gives you your, the NPK. But the idea that once we get a soil biological analysis of that, the production area, then we look at what we have to do to maximize that. And if we have a little bit of work ahead of us, then we're going to be dependent upon that supplemental fertilizer until we get that all snapped into place. And it could be a year, or it could be 6 months. It could be, 2 months, all depending on, where things stand with the result of the test.
All right. Are there any edible mushrooms that work well with cannabis as the fungal portion of the soil?
That I would defer to. That I don't have that answer for you, so I would defer to a more specialized mycologist to give us that answer.
Yeah, that's fine. I was going to say that's kind of a more biology question anyway. So let's see, use of kelp tonics. Is the use of kelp tonics good in living soil systems? Is it worth using?
Yeah, kelp is great. In a short, I do a lot of presentations that go much longer than this, and I can get into a lot of detail. I use kelp in a lot of my projects. It is, kelp, especially the species Ascophyllum nodosum, that is harvested in the Canadian Maritimes and off the coast of Norway. Kelp is such a rapidly growing plant in the ocean, and it has a lot of cytokinins, amino acids, things that all of our land plants want. So kelp should be looked at as the ability to deliver soluble potassium to a plant. So it's a potassium source. A lot of the kelps are like 002 in analysis, but it's also a soil conditioner, and it's also microbial food. So it can be used by the plant. It can condition the soil, and it can stimulate microbial activity. So I'm actually working to develop a granular fertilizer product that kelp is actually combined with soy to bring the benefit of kelp into the picture.
Awesome. What are your thoughts on high calcium soil allowing more oxygen in the root zone because of the size of the calcium molecule?
Calcium is very critical in soil structure, which is holding the pore space for oxygen. So low calcium, soil structure is not as great, not as much oxygen. Yeah. The thing with calcium that, when we say high, a better word is optimum, to get optimum calcium so the biomass and the plant can both function properly. But remember, again, the calcium is salt. So if we get too much calcium salt in there, then we're going to go the wrong way. Also, remember that high calcium can lock up phosphorus. So you can impede phosphorus' ability to enter the root zone if calcium's too high. So rather than talk about high calcium levels and trying to shoot for high, we always should shoot for what is optimum. And that's based on cation exchange capacity. So every soil is different. So when you have CEC and you get that number, say the number is 14.5 milliequivalents to 100 grams, that means that soil can hold on to so much and only so much. And calcium is soluble, so if you get too much, you're just going to have salt running through the system. So that's the key there, is what the soil can hold. If you had a pH of 7, the soil could only hold half as much calcium. If I had a pH of 25, it could hold 75% more calcium. So it's all relative. Every soil, it's all in relationship to CEC. Calcium should be looked at.
All right, great. What's your stance on top dressing with things like azomite, glacial rock dust to add trace minerals to the soil?
Yeah, absolutely. If you have a soil test that show that that certain minerals are needed, absolutely. Yes, mineralization and remineralization of soils, going back to some of the science from 100 years ago, that show that, that's a very valuable thing, but it's not something you do over and over and over again. Again, it's based on testing and what you have, and then if amendments are needed to boost it up to the optimum range, then yes, either of those two would be a very valuable input as an amendment. They would fall into the amendment category.
All right. We've got basically two questions here that are pretty similar, just about where should we have soil tested. I know you mentioned a couple of biological laboratories, but I think people are looking for, a couple more recommendations on labs, especially for chemical soil tests, and especially ones that might have recommendations for cannabis specifically.
I don't. The different labs make different recommendations. I don't know specifically labs that are geared towards cannabis recommendations. It's at this point, I think it's more each of us understanding what we know the cannabis plant needs and being able to interpret those recommendations. Out on the West Coast, I use Waypoint Analytical in Modesto, California. It's a one-stop lab. It'll test everything from my water to soils, to tissue, salinity, whatever I need. It is a strong acid laboratory, a Malic 3 extraction. One of the things with labs, I will caution you, whatever lab you pick, stay with it. Don't bounce around from laboratory to laboratory, because they use different methodologies of extraction. And if they use a strong acid, they extract a lot. That might be the total. If they use a weak acid, they extract a little bit, and that may be what's readily available, but not the total. So Waypoint Analytical that I use, I come back to Midwest Labs in Omaha, Nebraska, if I want more organic recommendations. So it's different all over what you should always use a laboratory other than the biologicals, because there's only really two good biological laboratories right now in the country, I believe. And one is in Corvallis and the other is back here on Long Island. But for chemistry testing, always use a laboratory in your region, because they have developed the acid methodologies for soils in that region.
Okay. And, along those lines of laboratory soil testing, I mean, in terms of recommendations, just maybe, what are your thoughts there? I mean, my impression is, recommendations you're going to get are probably going to be pretty conventional, right? Like, oh, you've got a deficiency of this, add some more of this, so we're trying to teach you, what do you do with that? That's not conventional, right?
Yep. That's exactly right. And that's why I don't ever use laboratory recommendations, because they are 100%, coming back from extension as conventional chemical. Yeah. And, here in Massachusetts, we've been, hounding organic now for 25 years. And, the laboratory will finally put in a piece of paper that says, there are organic alternatives. If you want information, call the lab. But when you get whether it's Waypoint or A&L, Logan Labs, any one of them, any of the university, extension labs, all recommendations will come back with conventional materials. So that's why in the organic industry, and Leighton and I have talked about that, we're actually working to try to create a body of information that we can share with cannabis growers on, if the soil test says this, what do I do organically as a recommendation? Because the laboratories will not give it to us that way.
Yeah. And how do we know if those numbers come back? Is there a way to know if you're getting total nutrients versus like just plant available nutrients?
Yes, there is. And that is by the testing methodology used by the laboratory. And, again, in the interest of time, because I tried to cover so many things, normally I talk for 45 minutes just on soil testing. So when we test soils, just very quickly, as I said, it goes to a laboratory. They put an acid in there. Malic 3 extraction is a strong acid extraction. And it will give you a big number for phosphorus, or say it gives me a number of 100 parts per million phosphorus, and that comes back high. And that's a Malic 3 strong acid extraction. That's all the phosphorus, basically, that's there in the soil for me to make use of, but it's not all available to the plant. A modified Morgan test is a weak acid extraction, where, stands to reason, the weaker the acid, the less soluble nutrients you get. So many people think that the modified Morgan is more similar to what the plant has to play with on any given day. So you're not extracting the total number, but you're extracting a portion of it. So phosphorus in Malic 3, 35 parts per million could be high. So same soil, same phosphorus, two different laboratories, two different acid extraction methodologies, and high can be 35 on one test and 100 on the other test. So then that's where it becomes, that's sort of the follow-up to a course like this. When I do my teaching in, land management and turf and landscape, what I've just done for the last 2 hours then gets followed by, here's the next steps on how we, and that could be something to develop here. The next steps where we all get together and we talk about how do we take that more in depth, take that soil test, and how do we make it work for us?
Great. Well, I think that's a great place to stop. We are just at the end of the session here. So thanks again so much, Chip, for taking the time and answering all the
questions. You're very welcome. Thanks for having me. I enjoyed it. Thank you.
Yeah, awesome. So that concludes day one of our academy here. That was quite a marathon of information. I'm sure everybody is tired and overwhelmed, and also hopefully super happy with everything they learned today. So we will see you again tomorrow, starting at 9 o'clock Pacific. First session is, I believe, with Kevin Jodry, talking about THC growing with living soil. So make sure that you locate your email that has the link to the whole event and go in and find the session with Kevin Jodry specifically. Usually you have to scroll down a little bit. I know some people have had issues with that. But email us if you have any other questions. And I think that's it. We'll see you tomorrow.